have a window, we kind of detect almost intuitively when we are in lighting conditions that are not good for us, that are kind of not appreciated or too dim and too bright. And so we have an understanding of what it means to be in bad lighting. And the research over the last couple of decades really has highlighted what happens when we are in the wrong type of light at the wrong type of time. And so my work really has focused on understanding the neurobiological pathways that are involved in controlling these aspects of human physiology. So all of the light reception that is relevant for us that we care about in this specific area of research go through the eye, and the eye contains specific cells that are sensitive to light. Now, of course, there's other actions of light that we shouldn't forget. So, for example, the impact of light, specifically sunlight or daylight, on vitamin D production. Here, we're only talking about the light, the ocular light exposure, and its then ensuing neurobiological effects. So what are the light-sensitive elements that we have? We have the rods and we have the cones. And really, until maybe about 25 years ago, the rods and the cones were the only known photoreceptors in the human eye and the human retina. The rods are sensitive to very dim light, and they help us see rudimentarily during dim light conditions. And the cones help us see during daylight. And, are the photoreceptors that are probably underlying your current vision and visual perception. There's 3 types of cones that give us color vision, and they're generally very well tuned to capture aspects of color, motion, and spatial detail. In the late 1990s, another class of photoreceptors was discovered, and so these are the so-called intrinsically photosensitive retinal ganglion cells. So the cones and the rods, they take signals, they kind of transfer them to secondary neurons or tertiary neurons in the retina that are called the retinal ganglion cells, which signals are summed up. And some of these retinal ganglion cells happen to be light sensitive themselves without any rod and cone input. So the way you can think about this is got the rods and the cones. They're the primary conduits, or the primary photoreceptor cells. And then in addition to those, you also have the special population of retinal ganglion cells that are sensitive to short wavelength light. And what we see is light or the signals from the rods and the cones are primarily projecting to areas that are involved in our vision, our visual perception. So primary visual cortex and the occipital lobe, which we call the canonical visual pathway. So right now, when you're reading or when you're seeing something, this all goes to the canonical visual pathways. The primarily IPIGC or melanopsin-mediated pathway goes a different route. They go to the hypothalamus. And specifically in the hypothalamus, there's a cluster of cells, a nucleus called the suprachiasmatic nucleus that is about the size of a grain of rice, that takes information from the eyes and then encodes basically or sends the signal to all other clocks in the body what time of day it is. And the neurons in the SCN are controlled by light, and of course they also have an intrinsic oscillation. And so what I've been working on for the last decade or so is understanding how different photoreceptors in the eye contribute to the different pathways. And so fundamentally, this is a research program that is focused around understanding the underlying neurobiology, but it also offers some insights into how we should use light in the built environment. And I think this is where it gets really exciting. The reason for this is, of course, when we think about what light we should expose to, we need to understand how light is processed in the retina and downstream from the retina and the brain. So to me, it's really key to bring this understanding of neurobiology together with, well, here's some dials I can turn or some free parameters that I have in creating spaces for people. So, and I want to give you a little bit of an insight, a little bit of a flavor and taste of what we do in the lab and how we study these things. Now, my work focuses exclusively on humans, so we generate data that comes from human participants that are volunteering in our lab. And of course, this necessitates a specific set of methods to be able to study in clinical studies, human responses to light. If I was working in an animal model, I could use different techniques. And I just want to tell you a little bit about how this might go if I was working in animal studies. So in animal studies, you have various ways that you could look at, for example, an animal that is missing specific photoreceptors. So you can generate an animal, you can breed an animal that lacks, for example, the cones and the rods, and you can ask the question, what function is preserved when you don't have the cones and the rods? You can do the flip side. You can make an animal that is lacking the iPIGCs, the melanopsin-containing retinal ganglion cells, and you can ask the question, which function is deficient in the absence of melanopsin? Of course, in humans, neither of these techniques are available, right? So we need to be a little bit more inventive, a little bit more creative in asking the question, how do the different photoreceptors contribute to different types of functions? So this is the human retina, now a different view. You have the three types of cones. They differ in what wavelengths they prefer. And there's the rods. And the rods really help us see only very coarse spatial detail at night, as well as the iPIGCs. Now, what's interesting for us is that the different photoreceptors in the eye prefer different wavelengths. And so here on the x-axis, I'm showing you the wavelength of the light. And you might have, if you've worked with spectral power distributions, for example, you'll have seen data also on this axis. On the y-axis, I'm showing you the relative sensitivity. So if this number is higher, it means that photoreceptor will be more sensitive to light of that wavelength. And so what you can see here is, interestingly, all of these photoreceptors, of course, this is what we call the visible spectrum here, from 380 nanometers to 780 nanometers. They are pretty much overlapping, right? So light of a given wavelength doesn't just stimulate one photoreceptor, it stimulates all photoreceptors, but differentially. And so what that means is that if we, as experimentalists, when we want to bring people to the laboratory and ask, hey, what do the iPIGCs do? What do the cones do? We cannot just use single wavelength lights. And the reason for this is, numerically, when we take, for example, a light that has the most of the radiance, most of the power, most of the photons at the peak sensitivity of melanopsin, it will co-activate all the other photoreceptors. And you can see this quite nicely here. You take a light that's short wavelength at around 490 nanometers or 485, you see that all photoreceptors are co-activated. So this is true for any light, right? If you expose yourself to daylight, it's got a broad spectrum. LED light has a broad spectrum as well, but differs a little bit in the shape. These lights are generated so that they illuminate the scene and don't specifically prefer any specific wavelength. Of course, when we want to study what happens when we shine a light into a retina of a person, we need to have a little bit more granular control. And so this is a very important principle in photobiology, or the biochemistry of vision, which is called the principle of univariance. So the principle of univariance, I'll walk you through it. It's not too straightforward to understand, but it's really key for understanding, how do we use light and how can we quantify the effects of light. So here what I'm showing you is the wavelength preference, what we call the spectral sensitivity of just one photoreceptor. And so I can generate three lights, I'm calling them E1, E2, and E3, that have different wavelength and different intensity, but actually produce the same response in the photoreceptor. So it turns out the photoreceptor, the only thing it can do is catch photons and convert that into some electrical energy, right? Some neural impulse, some current difference, some signal, right? And the only way it can do this, it can either go up or it can go down. And it turns out that when you shine a light on it, the information about whether it was a specific wavelength or a specific intensity is lost. So this is why it's called univariant. It's only changing in one dimension. So one single photoreceptor cannot tell a change in color or a change in intensity. This is also why if you're late at night, it's dim, you cannot see colors because you have only one photoreceptor, namely the rods. So you need multiple photoreceptors to be able to distinguish changes in intensity and wave. This was formulated by a Cambridge physiologist called Rushton, and it's really the basis for a lot of the work, and also the basis really for any metrology that takes light spectrum and converts it even into things like illuminance. Because what you'll find is there's infinitely many spectra that could correspond to 100 lux, right? Because, the weighting of light by the spectral sensitivity curve discards information about the spectrum. We can exploit this principle in experiments through a protocol called wavelength exchange. We can pick two lights, for example, that are matched in the S-cone excitation, here shown schematically. And when you alternate between this monochromatic light and this light, you will have no change in the S-cones, but a differential increase in the activation of melanopsin. So now we have a principle that we can use to basically examine how do the different photoreceptors, when we stimulate them in isolation, lead to different functions. We call this stimulated or silent, and in fact, we can do this for all photoreceptors, and so we can generate or manipulate spectra in such a way that when you alternate between them, you have basically you stimulate one photoreceptor class in isolation, but not the other. So here, for example, we've generated with a spectrally tunable light source a situation that stimulates melanopsin in activation, renders the LNS cone silent, and has a little bit of contrast on the rods because under daylight conditions, we consider the rod to be saturated. These classes of stimuli, right, that look the same to the cones or the same in chromaticity or illuminance are also called metameric lights. So a metameric light is a light that basically has the same pattern of activation on our visual apparatus. So they would appear the same in brightness and color, but have difference or yield to differences in the melanopsin system. It's also the reason why you could have the same color and illumination, right? The same illuminance and CCT, for example, with LED lights or with fluorescent lights or from a monitor, right? The human visual system discards the information about the spectrum and only takes kind of the reduced version of the spectrum. So I want to walk through a couple of studies that we ran that really tried to nail down how do the different photoreceptors lead to differences in physiological response. And I'll start with one study that I did early on in my career, which looked at the pupillary light reflex. So pupillary light reflex is a very easy and convenient method to examine how light impacts us. And here, so interestingly, this kind of video either makes people interested in this research or propels them away from it because it's kind of a creepy visualization of many different eyes. So the pupillary light reflex is basically measured using glasses that have video cameras. And what we can do with it is basically track how different light scenarios change the size of the pupil. And so in our setup, what we can do is we can stimulate one eye and then measure the pupil in the other eye. And that is because the pupillary light reflex as a biomarker is yoked, right? So there's a consensual pupillary light reflex. When you stimulate one eye, the other eye will respond. And so we can use that principle then to study. We stimulate one eye and we observe how do the photoreceptors in that eye lead to differences in response. So what we typically do is we then take our stimuli, we record pupil responses over many trials, and then we average across trials. Here what we did is we had a very slow modulation of the contrast of the photoreceptor, and we asked the question, does the pupil, so at 0.1 hertz, right, that's a 10-second cycle, contrast is going up, contrast is going down, how does the pupil track these changes in light? And it turns out we can then do some mathematical fitting to these data and then extract some interesting parameters. The first one is how much does the pupil respond? And the second thing is with what temporal relationship? So does the pupil immediately constrict or is there a delay? And so we did this across many sessions, many trials. This is about 70 hours worth of data for one participant. So looking at the same stimulus, and because the pupil can be quite noisy, you have to repeat the experiment. And so let's just look at one. So here what I'm showing you is just one cycle for a 0.05 hertz modulation. So that's 20 seconds. And so here what we see is a very slow increase in contrast, a very slow decrease. And as you would expect, when you increase the light, the pupil constricts. And when you decrease the light, the pupil dilates. And so we can summarize these types of data and plots that look like this. So if there's engineers amongst you, this is a plot that you might have seen where we plot in this kind of polar plot on the as the radial eccentricity is how much did the pupil respond, and then the angle around the circle corresponds to the temporal relationship. So what we nicely see is we cluster at 0.05 hertz all of the L plus M responses, so the responses to the cone stimulation, cluster in one quadrant, melanopsin clusters in another. And now the question is, where do the S cones lie? And so for this, interestingly, as neuroscientists, we looked at the literature, and what we found was that indeed from animal work, there had been the finding that there's inhibitory S cone inputs into the IPRGCs. So what does that mean? So an inhibition in neuroscience basically means if you activate something, it reduces the activation of something else. And so here what was happening is S cones led to a decrease in firing in these neurons. So you increase the S cone drive, but in fact, even though there's more signal, it flips the sign and it reduces, it inhibits the firing of the IPRGCs. And so this is not just some interesting thing about neurons, because it actually leads to an interesting insight about pupil control, because it turns out that S cones, so the activation of the S cones, lead to an opponent response in the pupil. So what does that mean? When you increase the activation of only the S cones in your retina, your pupil will dilate. And that is different to all other photoreceptors where you increase how much they're activated and the pupil constricts, right? If you're in a dark room, you got big pupils, you turn the light on, pupil constricts. Except if you have lights that you can only generate in the laboratory, which stimulate the S cones. And so this is a very, very interesting property. It turns out to be true at different temporal frequencies. So in this paradigm that we use, we were able to find solid evidence for S cone signals that are actually dilating the pupil rather than constricting it. In addition, we also asked, well, depending on how fast the light is changing, how does the pupil size change? And this is not only of theoretical interest, but also of interest to understanding how much the fast variations in light influence our physiology. And here the insight is that, if you're sitting in front of a computer or you're sitting in a room, the light is probably not changing very much. If you're watching a movie, you have a lot of temporal variation. And if you're walking around outside, right? If I imagine stepping outside of my office into the parking lot, of course, suddenly I'll have a lot more illumination compared to the maybe 400 lux or I think 200 lux that I have here. So ultimately, this helps us understand how does our physiology change its response depending on how fast the light is changing. And you move your eyes, you move your head, you move your body. All of this leads to not a clean kind of dark light cycle, but actually something that's very complicated in space and time. And so what we did here is we measured, for slow modulations or for very fast modulations, how fast can the pupil track these changes. And it turns out for luminance, for the cones, you have a lot faster responses that the pupil can encode, whereas for the melanopsin, the S cones, it really prefers the slow changes in light. And this really tells us something about how we also should think about, how light should be used in the built environment, because ultimately it discards all of the high frequency information largely. And what matters is overall in time, what is the intensity? You see this isn't for one subject, you see it for another participant as well. We also pushed this idea of doing what we call the science substitution a little bit more. We developed a modified projector system that we could use to stimulate in space and spectrum separately. And basically we examined when we hide in these monochrome grayscale videos modulations that stimulate only the melanopsin system, what is the pupil response? It turns out you can easily do that. So in the pupillary light reflex, we find nice contributions of LMS cones and melanopsin. I'll walk a little bit through the next one as well, which also was very interesting. So what the one question that we had was, do the melanopsin signals also contribute to how we see the world? Does it contribute to our brightness, our color perception? And so what we did was we followed a neuroscientific approach in which we asked, can we measure signals in the brain? So if you've been ever in neurology department or radiology department looking at brain scans. You bring people in the scanner. They take high-resolution images of your brain. What we can examine here is how much blood flow goes to a specific region when we stimulate the eye with a specific photoreceptor. And so it turns out that when we do this, we could see that melanopsin signals, so the ones that normally only are involved in circadian rhythms or in melatonin production, also seem to play a role in our vision and our visual perception, which is quite interesting because typically we think that these melanopsin systems don't do a lot to our visual system. So what we could see is we see a melanopsin signal in primary visual cortex, in the occipital lobe of the brain, in the back of your brain, and we see also cone signals. I didn't show you the data here, but of course, because they're involved in vision, the cones, we also see these signals in the back of the brain. I want to switch gears a little bit and talk about melatonin suppression. So the pupillary light reflex and primary visual cortex are both visual functions, right? They're both related to how we see, how we perceive, etc. Except when we're looking at the effects of light, we also need to look at then the more hormonal, the neuroendocrine effects of light. And so this is something that sometimes is summarized under non-visual or non-image forming. This includes things like melatonin suppression, circadian phase shifting, and sleep. And so now about 45 years ago, Al Louis published a paper in Science that showed that light exposure in the night will suppress the production of melatonin, right? So the study of light suppression of melatonin only dates back to a little bit, less than 50 years. But really is something that is still going on, right? So this is where a lot of the research is happening. How do different lighting scenarios cause different secretion of melatonin? So here at the top, you can see what happens when you don't have light, which is just, the melatonin stays at a relatively high, stable level. But then when you suppress melatonin, when you put the lights on at night between 2 a.m. and 4 a.m., you see a nice suppression then of the melatonin. And we can use this as an assay to understand this pathway that connects the retina to the hypothalamus. And again, here's a plot that we had seen, wavelength across sensitivity. And so you can see quite nicely here the suppression of melatonin by different wavelengths, with the short wavelength light taking the precedence. And so what we asked was, well, what about the S-cones? We had just at the time found that the S-cones have this paradoxical response in the pupil. And so we wanted to ask, do we also see an influence of the S-cones in melatonin suppression? And so we used the same trick again, where we generated lights that had a strong difference in the S-cones, but no difference in the other photoreceptors, and put people in front of it and measured their melatonin through saliva. So these are the two conditions. A little bit of orange, a little bit of pink, and massive difference in the amount of S-cone stimulation. So it turns out there's actually no effect that we were able to see. So the S-cones, even though there was almost a 100-fold difference in S-cone activation, didn't produce any effect on melatonin or on sleepiness or on alertness. So we don't find an evidence for an S-cone effect, which basically cements the idea that the primary photoreceptor that is involved are the melanopsin-containing retinal ganglion cells. And so even though this is a negative effect, it also has a very important consequence, and that is when we want to evaluate what influence light has on our physiology, we only need to look at the IPRGCs. We only need to look at the melanopsin signals and not at the cones or the rods. So we do have a little bit of a positive data here as well, right? So people preferred the room light compared to the active light. They found the light more glary. And interestingly, we also see this slight drop in general well-being in our experiment, right? So these are long experiments in the evening, and then a slight uptick, which we call the going home effect. So participants are in the lab, very happy then to finally leave the lab after 6 hours of giving repeated saliva samples. So melatonin suppression looks like there's no signals from the S-cones, but rather the signal that we see is driven primarily by melatonin. I briefly want to talk about one group of people that we studied, and that is people without cones. So it turns out there's some folks that have conditions that renders their cones dysfunctional, right? So these folks are quite debilitated by light. They have light avoidance and also typically wear sunglasses or other glasses to kind of drop down the intensity of illumination. And so we had a population of them in the lab. And so this is called congenital achromatopsia. This is a genetic defect. And so we tried to find in prior literature whether there's any references to sleep. The only reference that we could find was a New York Times article that said that many with the disorder are proud night owls who love going out after dark. And of course, if you're very sensitive to daylight, then this is exactly what you would do. So we threw a couple of things at this patient group, measuring visual discomfort, but also tracked the melatonin and also did genetic confirmation. And so it turns out when you don't have the cones and when you don't expose yourself to daylight, you nonetheless seem to have normal rest activity cycles. Similarly, in the participants that we were able to measure melatonin in, we were finding normal melatonin production patterns. And so this would seem to suggest that when you don't have cones, there might be some adaptive processes that nonetheless lead you to have a normal circadian property. And then finally, I want to talk about one study also from the lab where we looked at how light changes during twilight. And so it turns out that, of course, and we all experience this, when the sun goes down, the illumination of the Earth's surface, of course, reduces quite drastically, right? So we see a drop in about 5 to 6 orders of magnitude between the sun being 12 degrees above or 12 degrees below the horizon. At the same time, and if you're a photographer, I think you might appreciate this, there's a shift in the color of the light, and that is called the blue hour. So we see a shift in the relative spectrum that boosts short wavelength light just around dawn and dusk. And so now the question is, well, is it that our retina, our eyes, encode aspects of this twilight in some specific way? So what we did in this study, and this was led by Christine Blume, this is not work that is published, we again used the same trick to generate lights that are biased along the blue-yellow axis and ask the question, do these have any specific influence on the circadian system? So we used a protocol that is quite involved, where people came to the lab for one adaptation night to kind of get used to the lab, and then visited the lab 3 times in total for about 32.5 hours in every session. So it's a lot of hours that they spend in the laboratory. We exposed them to a light, and then we asked, how much did this light shift your circadian rhythm? So it turns out that there was no difference between our background condition, which was just neutral light that was constant, and then stimuli that differed in how much they activated the blue-yellow axis of human vision. No difference in circadian shift, no difference in melatonin, but of course you see a suppression of melatonin by our stimulus. We also didn't see any differences in sleepiness or in the alertness. And so what this would again suggest is that indeed the primary photoreceptors that are underlying the control of our circadian clock are not the cones, but it's melanopsin. And so even though there's a lot of negative results here, it really cements the idea that when we try to evaluate light, and when we want to know how does light affect our physiology, we don't need to look at all 5 photoreceptors, but just melanopsin. And so this simplifies measuring light, planning buildings, evaluating light exposure quite a bit. I want to zoom out a little bit and tell you a little bit about what we've been doing and where it kind of connects to the real world and ultimately to policy. So most of the experiments that I've talked about really talk about biological capacity. We've done changes in light exposure that are quite unnatural, that are parametric, so we can understand under these isolated conditions, what is the impact of the light. And so, of course, when people come to the laboratory, we control what they do, we control their exposure, and all of this helps us understand what are the biological mechanisms. So we're really probing how far can we push the system to understand, do the estrons make a difference? Is it melanopsin? Is it something else? Now, the real world is, of course, very different, right? The real world is messy, it's noisy. From a measurement perspective, it's really hard to know exactly what the exposure is that I'm getting. And it's also very hard to measure what is the physiological influence, because in the lab we can control what people eat, in the real world, we know that, for example, citrus fruits or bananas influence our melatonin. So if you do this in the field, it's just quite tough to get clean measurements. And importantly, many things can influence us, right? Exercise, nutrition, how we slept, right? And so the real world relevance, right, and the biological capacity are really tough to square up. And importantly, behavior, the light exposure we select also makes a huge difference. I think that, overall as a research program, it's worth trying to put these together. And one way to do this is to do what we call, or to follow a research program that uses what we call physiologically relevant measurements. So we can control, we can characterize mechanisms in the lab, and we should use knowledge of these mechanisms to evaluate what is happening in the real world. And so you might be aware that 2 years ago, Tim Brown and Ken Wright led a group of international experts to develop light level recommendations. And so I was part of that group. It was a really, really great exercise to think about what do we know about how light affects us and what are the light levels that we should reach. And this is, of course, where for the built environment, these are targets that I think we should keep in mind. So, and this effort looked at many different outcomes, looked at melatonin suppression, it looked at circadian phase shifting, and then also KSS, which is the sleepiness scale. And so putting all these together then basically yields a series of insights, a series of curves that basically say we have recommended light levels for daytime indoors, pre-sleep, and the sleep environment. And so the light levels here are 250 lux melanopic EDI. So this is not the lux that you might know from a lux meter, but it is the melanopic intensity of daylight that has the luminance of 250 lux. For the pre-sleep environment, we recommend less than 10, and for the sleep environment, less than 1 lux melanopic EDI. And of course, now the question is, well, these are great values. How do we move this forward? And I think this is something that over the next years we'll have to figure out. What is the evidence base? How much can be trusted? What about individual differences? But I think it's an important step moving towards a scientifically based evidence base for making decisions about what light exposure we should get. We try to also think about what is the light exposure we get, not what we should get. And what we do is we develop this term called the spectral diet. And just like a nutritional diet, we think about the spectral diet as the totality of exposures that you get. And of course, in order to characterize this, we need light loggers. And so we and many other groups have been working on light loggers that help us understand what is the light exposure that we are getting. And so I have a prototype here next to me. This is the same one that you see here. We're just working on characterizing this and really then to try to understand what is the exposure that we get in the real world. We've also done some work that not only connects light exposure, but also physiological responses. So this is where we're really doing field research. Here we bundled up a spectrometer and an eye tracker, and we asked people to wear it. And so what you can easily see is there's differences between daylight, not only in the exposure you get, but also in how big the pupil is. So here we use the pupil as an index into the effect of light in the real world. This was work led by Raphael Lazar at the University of Basel. This paper is coming out in the next couple of weeks, where we looked at, in fact, pupil size across the lifespan. And so it turns out that how well your pupil responds to light depends on how old you are, right? So you have a phenomenon called senile meiosis, which basically says the older you are, the smaller your pupil. And so this is reflected here in these two curves from an 80-year-old or an 87-year-old individual in our study, where you see, of course, differences in, at the dimmest light level, how big is the pupil. And so this is all, for all participants, we see a very nice relationship between age and light illumination. This interaction basically between how much the pupil constricts with light. The other thing that we've been doing is collecting really a catalog of the types of scenes that we are exposed to. So this is work led by my PhD student, Nilüfer Tabande. We've collected a total of 850 or so scenes across the world, measuring not only the spectrum, but also using a multispectral camera that could help us collect the information about how much is a photoreceptor stimulated by a given scene. So not just integrating across space, but also giving per pixel in the field of view how much a photoreceptor is activated. And so we're starting to do some very interesting statistical analyses about this to understand what is the visual world that surrounds us made of. So I want to leave you with this because I think it is a very interesting story. This is about 10 years, a decade worth of work. And what you can see is that we found some things. We also found the absence of specific functions. I think this slide to me, I think is very interesting because it shows that there's still a lot more that we need to understand as neurobiologists or as neuroscientists to understand really what mechanisms do we have and how do they work. And I think it also shows that there's still a big gap, right, between the basic research that we have or that we can do and the applied research, right? So if you ask me, okay, well, you've shown that the S cones don't contribute to these functions. Does that mean they never contribute to these functions? And the answer is, well, probably you could do experiments in which you make them more sensitive and you try to boost them and maybe make them contribute to the function that we're looking at. I do think there's some recommendations that we can follow, and I think the light level paper that I showed you, the recommendations are a very great starting point. But of course, there's still, of course, a lot more work to do. And I think this is very exciting, and I want to kind of use this opportunity also to say that, it's not just understanding biological capacity, it's also, well, how do we make this knowledge count in the world, right? And this is, I think, ultimately where conversations like this or talks like this are very helpful to bring the knowledge that we have to the folks that build spaces and think about how do we optimize light. So with that, I want to, just two things. We do run a seminar series called the Non-Image Forming Effects of Light. This is free to attend every Monday at 5 p.m. So if you're interested in joining, it's on our lab website. And the other thing that we recently did was launch a podcast. So we have a science communication podcast looking at light and circadian rhythms. We just published the last episode of this season, with one more coming up, which is a video episode from listener questions. So that will be released in a couple of weeks.